Swayam Prabha. Digital India. Educated India.
So, welcome you all to the next class on inorganic chemistry of life principles and perspectives. In the previous class, I have tried to summarize the things relevant to the introductory aspect of that, and let me continue with some more highlight speaking to you. And in the introductory part, I also have talked about some techniques. Techniques based on the spectroscopy, techniques based on the microscopy, techniques based on the diffraction, and techniques based on the analytical kind of thing like mass spectrometry, etc. So, various things and lifetime measurements. So, a host of techniques are there. So, each of this technique is good in characterizing these enzymes. Not only characterizing these enzymes, when the enzymatic action goes on, you can also trap some intermediates, you can also study the mechanistic aspects, all of these. Things like mass spectrometry, absorption spectroscopy, EPR spectroscopy, fluorescence emission spectroscopy, CD spectroscopy, okay, and single crystal X-ray diffraction, protein, and many things like NMR relaxation times, fluorescence lifetime, and NMR spectroscopy including paramagnetism, extended X-ray absorption fine structure spectroscopy. Many of these things are there, and Mossbauer spectroscopy. So, all of these, and voltammetry is one of the very vital kind of a technique. Then you can also have some several protein based techniques like protein gel electrophoresis, where you can study not only the purity of the protein, you can also study the protein plus small molecule, a protein plus drug, a protein plus something else binding, how strongly they bind, a protein plus another protein, enzyme plus another protein, a nucleic acid plus protein. All these kinds of interactions, their binding strengths can also be studied, and proteins can be isolated, etc. If you look at even a simplest technique which we may think, circular dichroism, as you can see that you have features which are very characteristic of their alpha helical structures, beta sheet structures, etc. You can see from this figure for these, etc. This is just for as an example I am showing, but that does not mean that is only this one. And you know that protein techniques, there are a lot of protein techniques there, protein solubilizing, protein salting out. Salting it out means bringing the protein out and taking the protein in kind of things. So, the ionic charge, ion exchange chromatography, electrophoresis, isoelectric focusing, variety of things. So, these could be even based on the polarity based. So, that is their adsorption chromatography, paper chromatography, reverse phase chromatography, okay, hydrophobic, variety of these things. Based on the size or molecular weight, dialysis, ultrafiltration, gel electrophoresis, gel filtration, ultracentrifugation, variety of the techniques are there. So, you can do protein isolation, you can also do protein purification. These are all from the natural methods, but sometimes you may not have much of a production from the natural, then you have to do the genetic engineering and then express in the E. coli. So, that is kind of thing, binding by affinity chromatography, etc. we use in that. So, this is a little bit of extension of the chromatography equipment, looks like this. You have on one side the charge, that is plus, and the minus applied. So, between these two the flow will be happening. The flow of the proteins is because of their shape, because of their size, because there is charge, etc., etc. So, that based on this, you can in fact bring a correlation between the molecular weight and the logarithmic relationship between the molecular size of the protein and its the mobility, the electrophoretic mobility of these. So, these are logarithmic scales, people look at that. So, you can find various things there. Besides these things, we also have microscopy techniques where you can use scanning electron microscopy, transmission electron microscopy, atomic force microscopy. This can be used at a biomolecular level, this can be used at a cellular level, and you can see for the cell the SEM of the cell where it is showing all different parts of the cell, etc. you can identify. And you can also look at the beam here, example is shown for the TMF, the gold nanoparticle, and in fact you can coat a protein too nicely. And you can even see by atomic force microscopy, these are nothing but the DNA, and the DNA imaging is coming with in the atomic force microscopy. So, using this microscopy you can get the shape, you can get the size, you can get the height of the things in terms of the size, so many different factors, and when the enzyme substrate complex, when the enzyme regenerated, how the changes happen, all these things can be studied as well. And so based on this, that all these things, now we are at a stage where we can classify the metalloproteins and metalloenzymes. So, the metalloproteins and metalloenzymes can be doing simple hydrolase activities, or it could be doing peptidase activities, or they can be doing oxidoreductase, or they can be doing some kind of a transferring a group from one to the other, or they can be doing isomerizing a particular moiety or group or geometry, or they can be doing lyase, which is removing without the hydrolysis part. Hydrolysis can also remove. Ligase, take two parts of it and join together. So, overall the whole bioinorganic enzymes, biological inorganic enzymes, the kind of functions that they perform, these are the ones besides their storage, transport, etc. I guess you understand these. These are all various kinds of enzymes. So, what kind of things that we have already seen earlier, that the transition metals like vanadium, manganese, iron, cobalt, nickel, copper, zinc, molybdenum, all these things, they can do oxidoreductase properties, some of them can do electron transfer properties, some of them can do hydrolase properties, some of them can do lyase properties, some of them can do ligase properties. So, variety of these things, oxidation, reduction, as well as the hydrolysis, group transfer, isomerization, and the various kinds of things are this. Of course, in this electron transfer, proton transfer kind of things are also possible in all of these. So, this is overall that I have talked to you about the overall introductory aspects of the metal ions in biological systems. I have treated this is very well suited, even if a BSc second, third year student sits for this particular course, would be able to understand, because I have built the basics required for that, from the point of view of the metal ion elements in biology, from the point of view of the coordination chemistry, from the point of view of the biological molecules, also from the point of view of the techniques that are used. Now, let us smoothly transit into the biological inorganic chemistry of alkyl earths, and then followed by transition metals. So, let us look at that. I have talked to you about sodium, potassium, magnesium, calcium. They are very important. Sodium is very high outside, low inside. Potassium is very high inside and low outside. Therefore, this sodium and potassium have to be transported across the cell. Similarly, you have magnesium is quite good both in external, internal. Calcium is also there. The magnesium is found much more than the calcium inside the cell. Therefore, the calcium bound proteins have to be protected in some way, have to be special featured in some way, so that the magnesium ions will not trigger or activate the calcium proteins. Now, how it is doing? Yes, you know very well that the calcium proteins have a special affinity towards the carboxylic groups, carboxylate moieties. Therefore, some of the post translational proteins give additional carboxylate moieties too, and they all will make this one. So, overall, if you look at the in a very general fashion, the calcium binds to the protein mantle or protein molecules or proteins much higher, like a 10 power 6 range binding constant, 10 power 3 range for magnesium, 10 power 1 range for potassium, and 10 power minus 1 range for sodium. That means, between sodium and potassium, calcium, there is a 10 million fold difference. Between potassium and calcium, there is a million fold, 100,000 fold, a million fold, 100,000 fold, and 1,000 fold difference is there. So, magnesium of course activates kinases, phosphatases, mutases, and lyases, and many involved in many biosynthetic processes. The main thing is kinase and phosphatase absolutely. These are the two ones which are running the whole the signaling process and all the events that take place. In addition, calcium will show many of the properties plus the triggering of this. Specific feature I talked to you is called the GLA proteins, which are nothing but carboxylated ones. Gamma carboxyglutamic acid are the ones. Calcium has a special feature, unlike the sodium, potassium and magnesium, that when the calcium enters into the cell membrane, can be captured at the inner membrane and something else gets released over there, it is called secondary messenger. So, in various ways the calcium is released and then that will activate some other kinds of things too. It can even activate some phosphorylation as well too. So, calcium can act as not as a simple primary, it can also act as a secondary. This can also be very well seen from its stimulation between the neurons and muscular junctions. So, one is from the neuron, another is from the muscle. So, the neuron muscle at this particular junction, the signals will be carried over, and these are all fully responsible by the calcium ion concentrations, calcium released components, all these kinds of things. I hope we understand that. Okay, in the process I have also talked a lot of small molecules, how they bind, how their stability constants, how the transport characteristics, all of these. So, what we found from all of this is that the size of the pore of the ionophore, size of the ion is important, but not sufficient enough. Then the nature of the ligands present in the ionophore and the number of ligands are present in the ionophore, the outer surface of the ionophore, lipophilic, hydrophilic, all these kinds of things, how well it can cover the metal ion, all of these factors. So, we have seen all of them in the transport. They can be passive transport, they can be active transport. We have seen all of those parameters. Let us look at one highlight of the example here. One of these curves that shows here the sodium transport by a particular this molecule called 222 cryptand. So, that is 2 oxygens in the linker, another 2 oxygens in the other linker, 2 oxygens in the other linker besides the nitrogens, and this is called 222. In case of 222, if you use the sodium will have a very high binding greater than log case greater than 7. Potassium is having greater than 8, and the cesium is having the log case is around 4. So, if you see that the transport, the cesium is transporting maximum than the sodium than the potassium, you see that. And then if you look at the stabilities is sodium is greater than, the potassium is greater than the sodium greater than the cesium. So, therefore, these two are not directly proportional, it could be, you can say, reverse. Now, instead of that, you take 2 and one more is carbon chain, 8 carbon chain and no 2 oxygens. Then what will happen? So, then what will happen is the cesium shows very little, sodium shows somewhat better, potassium shows maximum. So, potassium has got a 5 log case and sodium has got 3.5 log case and the cesium has got 2.7. So, in this case the transport is greater than potassium is greater than sodium greater than cesium. Similarly, stability is also potassium is greater than sodium is greater than cesium. So, in one case it shows the stability and transport are inversely proportional. In another case this is seen as the stability and transport are inversely directly proportional. So, what we will take? We cannot take either of these. So, there is something else which I have brought to your notice. The highest, the optimal seem to be around 4 to 5, 4.5 to 6 is the log case. So, whenever you have the 4.5 to 6 log case, you get an optimal transport, a maximum transport with optimal stability. So, very low binding will lead to a disruption of the transport before it is reached the destination. Very strongly bound case, it will reach the destination, but will not be able to release the ion. So, therefore, either case it is very dangerous, very strong binding of the ion with the ion by the ionophore, very weak binding of the ion by the ionophore, both are dangerous. Whether it is an ionophore, whether it is a synthetic one or natural one, or it is a protein binding to that, because in some of the cases not only small molecular peptides, the protein will also involve in transport phenomena too. So, they will bind too. So, so that means when you are talking about the ATPase, you know that the ATPase is an enzyme which requires the ATP hydrolysis during the transport of the ions from the outside to inside of the sodium, from inside to the outside of the potassium, and in these two are triggered by the two conformational states of the protein, and these two transformational states are triggered by phosphorylation and dephosphorylation. And phosphorylation is activated when sodium is bound, dephosphorylation is activated when the potassium comes in close to that particular thing. So, therefore, that is how in the transport of this the protein has an affinity and core for sodium binding. In the other conformation, in the phosphorylated one, it has a potassium core and potassium affinity. So, therefore, the affinity changes. So, affinity of the protein towards binding to sodium or potassium switches over from sodium to potassium when you go from the ATPase enzyme in the normal to a phosphorylated enzyme, and then back, etc., etc. So, these are some of the things that I have talked in the context of the transport aspects of this enzyme. So, the transport aspects of the transition, the alkali and alkaline earth enzymes. Okay. So, now I think we have looked at a lot of aspects pertinent to the elements in biological system. So, elements in the biological system, their relevance to the coordination chemistry, their relevance to the proteins, their relevance to the stabilization, their relevance to the lability and the transport aspects, etc. we have tried to look at. Now, we will look at some highlights. The highlights is highlights of the transition elements, how these transition elements are involved in the enzymes. This is where we will look at the highlights. Now, what are the transition elements where the enzymes are present? Vanadium, manganese, iron, cobalt, nickel, copper, zinc and molybdenum. Then we also look at some on the mercury and some on the selenium. So, this comes from the cysteine, selenocysteine, and this is the mercury reductase. So, removal of the mercury from the environment. It could be even from systems as well too. Okay, coming to the first system, which is the vanadium. Vanadium, as we know, can have oxidation states from -1 to +5 in the chemical systems, but the oxidation states which are very important or are possible in the biological systems are +5, +4, +3. Also explain to you why not the lower like +2, etc. You know, the +2 is a strongly reducing, it will easily go back to the oxidized one, and the many things below as well, they are not stable. So, therefore, +2 and below are not found, +3, +4, +5, and these are of course found, they are being identified in various organisms, and they do variety kind of functions. So, this is also boon in disguise. +5 can be studied by vanadium 51 NMR, +4 can be studied by the EPR, and things of this kind one can try to look at these ones. So, the things that we have explained to you was the phosphate can inhibit the vanadate can inhibit the phosphate in the phosphorylations in the sodium potassium phosphatase cycle. At the second half of the cycle where the potassium comes activates to remove the phosphate group. At that stage, if the vanadate is found, it will bind, it will be stopping the cycle of the ATPase. Therefore, the ATPase is completely inhibited. Therefore, ATPase inhibition is possible by vanadate. Now, that is the negative aspect of it. Let us look at the positive aspect of it. So, it is the peroxidase activity. It is actually called haloperoxidase. The example I have taken is a chloro. There can be chloroperoxidase, bromoperoxidase, iodoperoxidase as a kind of an enzymes. These are the enzymes which are found in algae, sea algae. These are the enzymes which are responsible for converting the organic molecules into halogenated organic molecules. So, in these cases the enzyme, the vanadium is found like a vanadate which is suspended in the enzyme, and one of the coordination is coming from the histidine and from the protein. This is the protein histidine. So, therefore, it takes more or less like a trigonal bipyramidal, more or less like that, and this is in vanadium 5 form. You can very nicely study the coordination sphere, coordination kind of a ligands using the 51 vanadium NMR. And this in presence of the H2O2 peroxide will try to interact with the peroxide, and then the peroxide species will be bound. This in presence of the halide can make break this one and form the hypohalous acid. This is the species which interacts with the substrate and give halogenated substrate, and then goes back to the normal. So, this HOCl is not just like that coming out, it comes out and then reacts with the substrate. So, that means substrate should be bound very close to this in the enzyme, and indeed, and then you have the kind of an enzymatic action going on. So, H2O2 addition, protonation of the bound peroxide, addition of the halide, and these are the kinds of things. So, overall what we have studied at that time when I talked to you the story of the vanadium. So, I have explained to you that the vanadium has been involved in variety of enzymes like haloperoxidases, nitrogenase, etc. Nitrogenase I have not explained because I will be explaining completely under the molybdenum. Also the vanadium nitrogenase details are not so very well known, but it is thought to be that the vanadium nitrogenase functions very similar to that of the molybdenum nitrogenase. Okay, different forms of the vanadates are present depending upon the pH and concentration in the whatever the biological medium or outside too. So, vanadates can bind at the place of the phosphate and cause the inhibition. So, I have explained the mechanism of haloperoxidase, and in fact, if there is a sulphate, the sulphate can do inhibition, and that is how this is being found in these kind of things. So, this is overall highlights of the vanadium, and let us look at some highlights of the manganese enzyme. Once you open the doors for manganese, you have a variety of things. It can act like a superoxide dismutase. It can be involved in the photosynthesis. In the photosynthesis, this is involved in the photosystem 2. What is the reaction of the photosystem 2? I am sure you are aware that it is going from water to oxygen. Water to oxygen, what is the reaction? Oxidation. And water to oxygen, how many oxidations? 4 electron, 4 proton oxidation. So, therefore, from water you have to get O2. That means you have to remove 4 electrons and you have to remove 4 protons, and that is where this is involved. I will explain you in a while. And catalase, what does catalase do? Catalase does, because SOD converts the O superoxide radicals into water and H2O2. That H2O2 is further converted by catalase into water completely, or oxygen kind of thing. And we have the ribonucleotide reductase too. Of course, we will be explaining more detail of the ribonucleotide reductase in the iron story now, and the dioxygenase. Dioxygenase will explain to you the more, we already explained you more details under the story of copper as well as under the story of the iron, but not in the story of the manganese. So, the main thing is explained is the SOD and the photosystem 2 and the catalase, but in the highlights I will take up the photosystem basically. So, what is the reaction that, as you can see in the light based reaction, okay, a dark reaction, CO2 getting reduced to carbohydrate, there is a lot of ATP involvement, and the water to oxygen, there is a lot of ADP involvement of all this. So much of ATP involvements of all this, as you can see, the light reactions, the dark reactions, and these are the coupled reactions that you have. So, these go at the photosystem 1, this will go at the photosystem 2. 2, and we as the bioinorganic chemist, inorganic biological chemist, we are interested in the photosystem 2 reaction of converting water to oxygen. And as you can see that these are the kind of light harvesting devices, and the electron transport system, and then you have the reactive center, which is the manganese. So, you have the light receiving, which is called antenna, electron transport, a huge transport system, and the reaction center, manganese center, where the reaction occurs. So, at the manganese center you can see here it is kind of a cubane, and there of the manganese oxygen alternate, except one of the thing is by the calcium, and that the fourth manganese is somewhere here, okay, and this is bridged by the kind of an oxo species this. Okay. Now, you see that. So, what is the kind of things that you require? 4 electrons and the 4 protons. See, this is S0 is what? Is a dark state. So, at the dark state is the S0 state of the enzyme, where 3 of the manganese 3 and 1 manganese 4, and this when the first pulse of the light falls on it, and then light drives this reaction and drives out a proton and activates to S1 state, and this S1 state will have the one of the manganese 4 is also converted to manganese 3 kind of a situation, because of reduction. So, one more electron, one more manganese 3, this is one more electron out, sorry, electron is out. So, 1 manganese 4, manganese 3 to manganese 4, then one more electron more, one more manganese 3 to one more manganese 4, like that. You have one more proton and electron, then the state comes into the manganese 4 system, or manganese 3 with 3 manganese 4. It is probably at this stage where the oxygen is evolved and the system is regenerated. So, overall you have. So, the protein goes through the S0, S1, S2, S3, S4 and with the 4 photon signals, 4 electrons, 4 protons, photon signals, etc., etc. So, what is that? You have the 4th manganese and the calcium, both are having water molecules. These are the 2 water molecules which are involved in the O2 evolution. That means 2 water molecules have to come closer, and so you have a water one over here and water one over there. So, there is a calcium 2 plus with the water, and then manganese with the water. Here you have OH and H. So, therefore, these are the ones where you have to have. From here, if you have to make O2, then OHO has to come, then OO bond has to form, then cut this bond, etc., and then finally becomes the O2. Okay. So, OH, so therefore, OH2 versus OH2. So, this will become OH and OH, and then becomes O2, 2 minus, then becomes O2, finally to O2. And these are the kinds of steps that you require in this kind of a thing. That is what is the 2. So, the rest of the 3 manganese are not directly involved in catalysis, and one of the manganese and one of the calcium, the water, 2 water molecules, as you can see that, and then with the first pulse electron they come closer, then the metal redox undergoes, it will become OHO, and then further oxidation will become manganese 4, and then you see manganese O with the OH still, and now this will further convert to the redox here to the OO bond. Now, once the OO bond is formed, you have to start breaking them MNO and here the CaO kind of a bond, basically MNO bond. If you start breaking this one, then you will get 1 electron from here, 1 electron from here, O2, then the O2 has come, and the O2 is released kind of thing. So, when the O2 is released, it will go back to the normal state. So, okay. So, we could see variety of these things very nicely, 2 water molecules, how they give O2. Okay, so in the manganese involved in a lot of enzymes like SOD, catalase, dioxygenase, photosystem 2. SOD case, it decomposes superoxide into the O2 and H2O2, and in that case what it does, it goes through manganese 2, manganese 3, back to manganese 2 2 to 3, 3 to 2 kind of a thing. So, that is what happens, and then whatever the H2O2 comes, that is being converted to H2 and O2 by the two manganese centers present in catalase. Of course, I have not shown in the highlights, but I have shown in the regular lecture that you can see that. So, in the photo system 2, just now I explained to you that the 4 manganese but 1 calcium, primarily mainly there is 1 calcium and manganese which are involved to bring the 2 waters close by, make the O bond, then O bond of that too. Okay, that is the kind of a thing that we have tried to look at these ones, and okay, let us get into the next aspect where I have taken, after the manganese, I have taken the iron in the bioinorganic chemistry. Probably what I will do is I will take up this highlights in the next class, because iron enzyme will have so many things, because iron is in the transport, iron in the non-heme, okay, iron is oxidative, reductive, etc. All of these are there. So, no problem, and what I will do is I will explain these ones in the next class, and as I have talked to you about the transport of the sodium potassium ions, then I have talked to you about the vanadium enzymes, then I have talked vanadium enzymes and vanadium haloperoxidases, etc., how the bromination, chlorination, iodination occurs at these things, how the phosphate and vanadate analogues will play a role in place of the other, and then I have explained to you the manganese in variety of enzymes with more details of the manganese in the photosystem 2, where the water releasing the oxygen. Of course, I will take up the iron enzymes and the rest of the enzyme highlights in the next class. Hopefully, in one next class I can try to explain all the highlights of this. Thank you very much.